All right, today we are diving into a real molecular detective story. We're going to try and decode the very first instructions that turn a single, lonely cell into a complex, living organism. How does life's blueprint actually get written?
I mean, it's one of the biggest questions in all of biology, right? You start with this tiny ball of cells, and somehow it knows. It just knows how to build a heart, a brain, the whole shebang. So what are those very first instructions for life?
To get to the bottom of this, scientists took a close look at the very first few days of a mouse embryo's life. Now, at this stage, it's really just a small cluster of cells. But don't let that fool you. It is already making these huge, life-altering decisions about what it's going to become. This is literally where the first lines of the blueprint are drawn.
Our investigation focuses on two main suspects. They're a pair of proteins, the technical term is transcription factors, that act like molecular architects. You can think of them as the general contractors on a building site. Their job is to read the DNA blueprint and decide which instructions get used and when.
Okay, first up, let's meet SOX2. This protein has this really unique structure that lets it act kind of like a molecular wedge. It literally binds to the DNA and helps pry it open just enough so that specific genes can be read. It's the key to switching on the most crucial parts of the genetic code.
And of course, SOX2 isn't working alone. Its partner in crime is another absolutely critical protein called Oct4. Now, the important thing to know about Oct4 is that it's on the scene even earlier than SOX2. It's already there, kind of waiting in the wings, setting the stage for all the big decisions that are about to come.
So the scene of our investigation, it's this tiny group of cells in the early embryo called the inner cell mass, or the ICM. These are the VIPs. These are the cells that hold this incredible power called pluripotency. And that just means they have the potential to become every single cell type in the entire body. Everything. It all starts here.
So how in the world do you figure out what these architects actually do? Well, the scientific method here was both simple and, frankly, brilliant. Researchers created mouse embryos that were missing either Oct4 or SOX2. And you can see the result right here in this image from the study. The bright green stain, that's highlighting the Oct4 protein in a normal, healthy embryo. But look at the knockout embryo on the right. It's completely gone.
So by taking out one of these key players, the researchers could just sit back and watch what went wrong. They collected just massive amounts of genetic and molecular data, basically gathering clues from the scene to piece together the real story of this essential partnership. Okay, now this slide right here visualizes the chaos that followed. Each one of those little dots represents the genetic state of an entire embryo. See how the normal ones in blue are all clustered neatly together? That means they're all following the same developmental plan, as they should. But the knockout embryos in red and green, they're scattered all over the place. Without their architects, these cells are completely lost. They're just veering way off the blueprint.
And believe it or not, the next clue was even more dramatic. The investigation dug into which exact genes were being turned on or off, and what they found was wild. The genes you need to actually build the embryo itself, they were shut down. And what's worse, genes for building the placenta, a totally different part, were mistakenly switched on. These cells weren't just lost, they were reading the wrong set of blueprints entirely.
So this led the scientists to zoom way in, right onto the DNA itself. They started focusing on these very specific DNA sequences where they knew Oct4 and SOX2 liked to work together. This little shared worksite is called an Oct-SOX motif. The best way to think about it is like a special docking station on the DNA that's reserved exclusively for this architectural duo.
And here it is, the smoking gun. This chart focuses only on those special OCT-SOX docking stations. And look, nearly every single dot is in that bottom left corner. That means these sites were completely shut down, unreadable, when either Oct4 or SOX2 was missing. This proved it. They don't just work in the same general area. They absolutely 100% depend on each other.
Okay, so let's step back for a second and connect all these dots. What does this scientific detective story truly reveal about those first moments of life? What is the fundamental job that this amazing duo is controlling? So, let's just quickly summarize the key takeaways here. We've learned that Oct4 and SOX2 are absolutely essential for kicking off pluripotency. They have to work together at those specific docking sites, the OCT-SOX motifs. And while Oct4 gets there early, it's the arrival of its partner, SOX2, that really acts as the trigger. Without this dynamic duo, the cells lose their identity and just start reading from the wrong playbook. And this model from the paper just gives us a beautiful, elegant summary of the whole thing. You can see Oct4 and SOX2 coming together right there on the DNA strand. And this partnership physically opens up that tightly wound DNA, which allows the right genes to finally be read. It's this precise, delicate cooperation that unlocks the entire blueprint for building a new life.
And this isn't just some abstract story about embryos. Understanding this fundamental partnership, well, it's at the very heart of stem cell science. By learning how to precisely control these master architects, we could open up just incredible possibilities for the future of regenerative medicine. Maybe, one day, using these exact same principles to repair and rebuild our own bodies.